Welcome to Winspharma. Today's topic is cell. What is a cell? Why we call a cell as a basic unit of life? For example, imagine you are looking into a tiniest and most hardworking and surprisingly organized factory in the world. That is called cell. So you call it as basic unit of life. Every living thing like giant elephant or ant and you half woke during morning glass made up of cells. There are different shapes and sizes. They may be in round shape, long, flat and star shaped even. Then what are its biological activity? They give structure and functional unit of life. The major cells are divided into two types. They are prokaryotes and eukaryotes. What are prokaryotes? Prokaryotes are they lack cell wall. For example, bacteria. Eukaryotes, they have well defined cell wall like plant and animals. They are divided by fusion. Here mitosis takes place and size is 1 to 10 mu mm, then here 10 to 100 mu mm, and here in special cases like plant cell contains cellulose and chloroplast which helps in photosynthesis process. Now let's discuss what are inside cell. Okay, what are cell organelles? First one is nucleus. Nucleus is the largest cellular organelle with nuclear membrane, nucleoplasm, nucleolus and DNA, which is chromatin. Eukaryotic DNA is associated with basic proteins called histones. Ante eukaryote DNA have associated with basic protein called histones. Then these form nucleosomes. Group of histones form nucleosomes and group of nucleosomes from chromatin. These are what is chromatin? Fibers of chromosomes. Human have 46 chromosomes in our body. What are its functions? What is the nucleus function? Controls all cell activities and contains DNA for inheritance. That means your future generation DNA is present. If you reproduce, then this DNA is transmitted to your child. Now let's see the diagram of nucleus. Here nucleus, this is nucleolus and this is whole is called nucleus, which consists of nucleus, nucleoplasm, liquid present in the nucleus and nuclear pore, and one pore will be present, and nuclear envelope which is membrane, nuclear membrane. Then coming to mitochondria. Mitochondria is a bean shaped double membrane structure. Inner membrane was folded into cristae. Here mitochondria is a bean shaped structure where outer membrane is smooth and rigid and inner membrane is folded. It is called cristae. Then center of cellular respiration. Mitochondria is responsible for cellular respiration. What is energy metabolism? What does it do? Energy metabolism, carbohydrates, lipids and amino acids, and it is also called as powerhouses of the cell. Its name, other name. Which is I said you before, double membrane, outer is smooth and envelope, inner is folded as cristae. Inner chamber of mitochondria is called matrix or mitosol, which consists of circular double stranded DNA or RNA and ribosomes. Then coming to endoplasmic reticulum. What is endoplasmic reticulum? It is a network of membrane enclosed spaces that extends through cytoplasm. There are two types of endoplasmic reticulum. That is rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is nothing but it is attached with ribosomes, and in smooth one you don't have ribosomes. These are factories for protein biosynthesis, protein synthesis. And smooth endoplasmic reticulum, it is lipid synthesis, and it is also important in calcium supplements to cell. Rough endoplasmic reticulum helps in transportation, and smooth endoplasmic reticulum helps in detoxification of the cell. Now let's see what is endoplasmic reticulum. Here the nucleus membrane, outer membrane forms here this structure, and it have ribosomes. This is called rough endoplasmic reticulum, and the small parts which are divided, which don't have ribosomes on its part, is called smooth endoplasmic reticulum. Rough endoplasmic reticulum plays in protein synthesis, and smooth endoplasmic reticulum lipid synthesis, helps in lipid synthesis. Okay, then coming to Golgi apparatus. In eukaryotic cell, that are clusters of membrane vesicles, form diatosomes, which forms Golgi apparatus. It gives structure and stack of flattened membrane bound sacs. These structure are stack of flattened membrane bound sacs. These newly synthesized proteins from Golgi apparatus, in Golgi apparatus, catalyze the addition of carbs, lipids to proteins. This chemical modification of proteins are necessary to transport of proteins across the plasma membrane. To Golgi apparatus transport the proteins, newly formed proteins to cell across the cell anamata. Then proteins and enzymes composed in Golgi apparatus are secreted through ATP signals, like digestive enzyme from pancreas, involved in membrane synthesis, like peroxisomes and lysosomes. What are lysosomes? Lysosomes are spherical vesicles filled with digestive enzymes. It is also called as digestive tract of cell, and maintain cellular compounds in dynamic state by dehydration, degradation and recycling process. What are peroxisomes? Peroxisomes are microbodies. These are single membrane, circular or oval in shape. Enzyme catalyze protect cell from toxic effects of H2O2. Then cell membrane. What is cell membrane? It is also called as plasma membrane, which is thin and flexible, which controls movements of substances in and out of the cell, maintains internal environment, cell communication and signaling. These are main points: cell communication and signaling, and movement of substances from in and out. In cell membrane we have two hydrophobic head and hydrophobic tail, and this total is called phospholipid bilayer because two layers are there. And you have many like transporters, specific transporters, channels, surface proteins, etc. on the cell membrane. Then coming to cytoplasm. What is cytoplasm? It is a jelly-like fluid which supports and protects the organelles in the cell. Site of many metabolic reactions. Where many metabolic reactions takes place means in cytoplasm. Next, what are vacuoles? Vacuoles are fluid-filled sacs, larger in plant cells. When compared to animal cells, plant cells have larger sized vacuoles. These stores water, nutrients and waste materials, and these maintain pressure in animal cell. Then coming to centrosome. What is centrosome? A pair of centrioles located near nucleus in animal cells, which helps in cell division. Cell division by mitosis. Okay. Then what is cytoskeleton? Cytoskeleton is in cellular matrix they contain cytosol, cytoplasm, mixture of enzymes and metabolites and salts, then which contains complex network of protein filaments like cytoplasmic filaments, which are used for support, shape, structure and organization of cell. For example, microtubules, actin filaments, intermediate filaments, which combine to form cytoskeleton. What are cytoskeleton? You have microtubules, microvilli. Microtubules are intermediate filament. Microtubules, you'll have like some microtubules, intermediate filaments, and actin filaments. Okay, these form cytoskeleton, which gives structure to the cell. This is all about cell and cell internal organs. Transport process across the membrane. Why transport process is needed across the membrane? Because to survive, the survival of the cell, transport is needed. Transport of substances across plasma membrane is essential for survival of the cell. If cell want to live, it should transport its material. Since the plasma membrane is selectively permeable, means it will select some of the substances to go out and come in. So transport of substances occur through certain mechanisms. Mainly transport is two types: active transport, passive transport. What is active transport? Movement of molecules from lower concentration to higher concentration by breakdown or hydrolysis of ATP is called active transport. What is passive transport? Passive transport is movement of chemical substances from higher concentration to lower concentration until it reaches equilibrium is called passive transport. It mainly occurs in gaseous liquid solutions. For example, transport of oxygen from lungs to blood and carbon dioxide from blood to lungs. These are mainly divided into three types: simple diffusion, facilitated diffusion, and osmosis. We'll later discuss about active transport. First, let's discuss about simple diffusion, facilitated diffusion, and osmosis. What is simple diffusion? Simple diffusion means it is nothing but higher concentration to lower concentration from cell membrane. Okay, then coming to facilitated diffusion. What is a facilitated diffusion? It helps in diffusion of large molecules that are unable to penetrate to semi-permeable membrane through transporter. If ఒకవేళ ఈ వాల్ నుంచి దీంట్లో నుంచి రాకపోవడం వల్ల it will take help of some transporters specific transporters or non-specific transporters. The macromolecules will take help of transporters like specific transporters or non-specific transporters. For example, specific transporters లో ఏమవుతుందంటే ఈ హయ్యర్ కాన్సన్ట్రేషన్ ఉన్న దగ్గర ఒక షేప్ ఫామ్ అయ్యి ఈ మ్యాక్రోమాలిక్యూల్ ఇక్కడ అటాచ్ అవుతుంది అనమాట. ఇక్కడ అటాచ్ అయిన తర్వాత దెన్ వాట్ హ్యాపెన్స్, దెన్ ఇట్ చేంజెస్, ఈ ట్రాన్స్పోర్టర్ ఈ షేప్ చేంజ్ చేసి లోవర్ కాన్సన్ట్రేషన్ సైడ్ వచ్చి ఇక్కడ దీన్ని వదిలేస్తుంది అనమాట. సో దిస్ ఈజ్ కాల్డ్ స్పెసిఫిక్ ట్రాన్స్పోర్టర్ facilitated diffusion. And in non-specific transporter, what happens here? No binding, but it allows macromolecules go through it anamata. Then I'll explain you briefly. Listen. Now, specific transporter, facilitated diffusion. Solute binds to the specific transporter on the membrane side that has higher concentration, and transporter changes its shape and released into the lower concentration. Then ikkada non-specific transporter lo solute moves through a non-specific transporter from higher concentration to lower concentration. That's it. Now coming to osmosis. Osmosis is only occurs when semi-permeable membrane only allows water to transport. అంటే వాటర్ లోపలికైనా రావచ్చు, బయటకైనా వెళ్ళవచ్చు అంతే. మిగతా ఏ సొల్యూట్స్ని బయటికి తీసుకెళ్ళదు, లోపల తీసుకెళ్ళదు అనమాట. Special type of diffusion that applies only on movement of solvent, occurs only when membrane are permeable to water and does not allow solutes. The force with which this diffusion occurs is called osmotic pressure. ఆస్మోటిక్ ప్రెషర్ వల్ల ఈ సొల్యూట్స్ అనేవి మూవ్ అవుతూ ఉంటాయి అన్నమాట సెల్ లోపలికి బయటికి. ఫర్ ఎగ్జాంపుల్ లెట్స్ సీ ఎఫెక్ట్ ఆఫ్ ఆస్మోసిస్ విత్ ఎగ్జాంపుల్ ఆఫ్ ఆర్బీసీ. ఆర్బీసీ వెన్ ప్లేస్డ్ ఇన్ ఐసోటోనిక్ సొల్యూషన్. ఐసోటోనిక్ సొల్యూషన్లో ఆర్బీసీ వేస్తే ఏమవుతుందంటే సాల్ట్ కాన్సన్ట్రేషన్ ఇన్సైడ్ ద ఆర్బీసీ ఈజ్ ఈక్వల్స్ టు సాల్ట్ కాన్సన్ట్రేషన్ అవుట్ సైడ్ ద ఆర్బీసీ. సో నో చేంజెస్ ఇన్ ఆర్బీసీ. నో చేంజెస్ ఇన్ ఆర్బీసీ. అది తీసుకెళ్ళి హైపోటోనిక్ సొల్యూషన్, వాటర్ కాన్సన్ట్రేషన్ ఎక్కువ ఉండి అండ్ వాటర్ కాన్సన్ట్రేషన్ హై outside the RBC and low water concentration inside the RBC. ఏమవుతుంది, then water moves into the cell and cell wall get rupture, then RBC dies. RBC rupture అవుద్ది. Here what happens, if RBC is placed in hypertonic solution, water concentration low outside the RBC and high water concentration inside the RBC, then here the water moves outside and cell wall sink, so RBC gets shrink. Okay, this is example of osmosis. Then coming to active transport. Active transport we have two types. What are active transport? Two types: primary active transport and secondary active transport. What is primary active transport? Primary active transport is energy is utilized by breakdown of ATP, that is 40% of energy is used. And for example sodium potassium ATP is here. Sodium potassium pump will help like movement of potassium outside the cell and inside allows the sodium. Here ATP will be used for energy release. Then coming to secondary active transport. Here what happens means one solute like mainly sodium which binds to the carrier molecule transport proteins that carries molecules increases its affinity for the substance to be transported. When both the sodium and the substance are bound simultaneously to the carrier molecule changes its shape, so both the substances are transported across the membrane at the same time. For example, what happens here, the sodium and other substance which want to pass through the cell membrane. Here what happens, the carrier molecule will increase the affinity of the substance to be transported. When both అంటే సోడియం ఉన్న ఏదైతే సబ్స్టెన్స్ పాస్ అవ్వాలనుకుంటుందో అది వెళ్ళి క్యారియర్ మాలిక్యూల్ని అటాచ్ అవ్వడం వల్ల ఆ క్యారియర్ మాలిక్యూల్ అటాచ్ అయిన తర్వాత అది దాని షేప్ చేంజ్ చేసి మళ్ళీ లోవర్ కాన్సన్ట్రేషన్కి తీసుకొస్తుంది అనమాట. సో this is called secondary active transport. Here energy is required. These are of three types. What are these three types? Symport, antiport, uniport. Uniport అంటే ఒకటే సబ్స్టెన్స్ it want to move into one direction. What is symport? That is co-transport, like sodium and glucose. Sodium and glucose enters the cell. Antiport, that is counter transport, that is one direction sodium, one direction calcium. One sodium will enter into the cell, either calcium will outside the cell, that is called antiport. Now coming to high energy compounds. What are high energy compounds? Some compounds in biological system which on hydrolysis yields energy. If a compound is if a compound breaks down, then energy is released. Then these are called high energy compounds. And based on how much energy it is giving, we have two types: high energy compound and low energy compounds. High energy compound substances which possesses sufficient free energy to liberate at least 7 calories per mole at pH 7. This is important point. if a substance is break down, then it releases almost 7 calories per mole at 7 pH, is considered as high energy compound. Most of the HEC contain phosphate group except acetyl CoA. Most of the high energy compounds contain phosphate group. For example, phosphophenyl pyruvate, phosphocreatine, etc. Based on the bond present, there are 5 types of high energy compounds. There are pyrophosphates like ATP and pyrophosphate. Remember this structure. Then acyl phosphates. Then for example carbamyl phosphate or acetyl phosphate, enol phosphates, phosphoenol pyruvate, thiol esters, trioesters like acetyl CoA and phosphogannans, phosphocreatine and phosphoarginine. These are phosphocreatine is present in vertebrae, skeletal system, heart and brain. Okay. Pyrophosphate, acyl phosphates, enol phosphate, thyroesters, trioesters, phosphogens. Then let's discuss about high energy bonds. What are high energy bonds? High energy bonds are these processes, the HEC processes acid anhydride bonds, mostly phosphoanhydride bonds. These are formed by condensation of two acidic groups. Anhydride bonds are formed by condensation of two acidic groups. When these bonds are hydrolyzed, energy liberates. I think what happened, Lippmann suggested to use the symbol like this. This is called squiggle to represent high energy bond. Like ATP is written as AMP, like adenine monophosphate and ATP means adenine triphosphate kada. So PPP, which have two anhydride bonds. These are anhydride bonds, two anhydride bonds. Okay. Then coming to biological significance of ATP. What is ATP? ATP is important source of energy and which is known as adenosine triphosphate. What is its structure? It has one adenine group and one ribose group which combines to form adenosine, and 3 phosphates, triphosphate, which have phosphoanhydride bond. This is representing phosphoanhydride bond, and where adenine and triphosphate are joining, this bond is called phosphoester bond. Okay, phosphoester bond. Then it contains one adenine group, one ribose sugar and a triphosphate. It is considered as HEC because high energy compound because it has two phosphoanhydride bond. These have two phosphoanhydride bond. Here on hydrolysis అంటే ఒకవేళ ATP break అయితే we will get almost 7.3 kilocalories per mole. Okay. Then what are functions of ATP? Functions. This is energy source, main energy source. ATP is a main carrier of energy that is used for cellular activity. ATP can be converted into ADP, adenine diphosphate. That means two phosphates. Then after here energy released. Then removal of one phosphate group gives 7.3 kilocalories, and this energy is used for all the reactions which takes place inside the cell. ADP can also form in then also ATP ADP ka marachu. ADP, then ADP can convert into ATP also. That is like this, we can represent in this way. It can move. Okay. Then coming to metabolism. Metabolism, for activation of many activities, ATP is required. For example, glycolysis, breakdown of glucose, and fat metabolism, ATP is required. Movement. The muscle contraction is regulated by ATP. It provides energy to allow muscle filaments to slide. It is also used in movements. Then active transport. Before we discussed transport of macromolecules like proteins, lipids, we need ATP. We need ATP. In DNA RNA synthesis also we use ATP. Signal detection. In signal detection also ATP is used. So ATP plays an important role in major functions. Thank you for watching.